couldn't get up this early, so don't make any weird faces or anything. So, I would first like to begin with introducing you to the coastal habitats in the Skagerrak Sea. It is a very diverse and complex area that is situated between the saline waters of the North Sea and the brackish waters of the Baltic Sea. This is a very popular study site for researchers, and the Norwegian coast especially is a favorite among ecologists. The shallow coastal zone is known to provide habitats that can sustain high numbers and production levels of fishes, which makes it a target area for both scientists and fisheries. There are many fish species in this area that can utilize multiple habitats, and therefore they're influenced by the structure of the seascape. The seagrass and seaweed habitats in this area are diversity hotspots, as they provide food and shelter for countless species. I have compiled some mugshots of the few most common species here. We have Atlantic cod, whiting, haddock, corking wrasse, and pollock. The composition of the habitats themselves are also unique, and the highly complex seascapes positively influence the abundance of the organisms that have the ability to utilize multiple kinds of habitats. But even less complex seascapes are conducive for juvenile assemblages in the summer, and they're still useful. These are thriving homes, but these habitats are also very sensitive to disturbances, such as trophic cascades and other impacts on the food web. An example would be the removal of a top predator, a larger predator that would allow the increase of smaller predators in shallow water seagrass areas, which can cause a cascading trophic level effect. And one of these larger predators is the Atlantic cod, which is one of the most prominent and economically important species in Norway due to its large offshore spawning populations. Their life history begins with spawning in early March, and the juveniles settle to the ocean floor when they're about 3 to 5 centimeters long in shallow sheltered areas. They can live as long as 12 years, but very few survive to even 5 years, which could explain why coastal populations mature much more quickly than other populations. The majority of the spawning stock biomass is cod aged 2 to 3 years, because those individuals 4 years or older have a much higher mortality rate. There's also been many studies that examined how temperature influences their habitat selection. Coastal cod have been genetically categorized into various subpopulations by many researchers, and these studies reveal important factors that influence the species as a whole. Coastal cod have a limited range of 30 kilometers or less, and there are multiple populations along the Skagerrak coast in Norway, with a saline gradient as a barrier. Coastal cod are highly sedentary, and older individuals are highly site attached. Offshore populations may influence the coastal populations over long distances, and the Skagerrak cod actually grow faster than northern and western cod, but slower than the ones in the North Sea. And recent work suggests that the cod in the Skagerrak and outer Oslo Fjord are made up by two coexisting but genomically unique ecotypes with potentially differing and persisting adaptations to their local environmental conditions. Oslo Fjord is not only used heavily by fish, but it's also used heavily by humans, both for commercial and recreational purposes. Trawling for northern shrimps and recreational fishing is very common, along with recreational activities such as boating. There are 2 national parks in the area, and multiple and consistent human activities will eventually lead to changes in the environment, both physically and biologically, and almost always end up impacting the organisms that live there. So it's very important to examine the extent of this impact, and one of the signs of this impact is changes in ranges and biodiversity that are linked to rising temperatures due to human activities. But overfishing definitely has to be one of the most impactful things humans do to the environment. Depletions of fish stocks has been categorized as one of the most serious threats to marine ecosystems worldwide, and overfishing has an indirect effect on seagrass survival. The removal of top predators, such as our cod, causes a top-down cascading effect with algae overrunning that seagrass habitat, which endangers juvenile cod. And this is important because of their recruitment phase. Recruitment is one of the most important stages in an ecological community. It influences the overall longevity of a population, so their habitats for recruits are just as important. The juvenile nursery habitats provide a source for the adult stocks, because the juveniles have to grow up and go somewhere. Habitat also has a significant effect on recruitment. Young juveniles are planktonic feeders, and they rely on eelgrass meadows and coastal areas to access food and safety from predators. Recruitment is mainly determined between late June and mid-August, when they've had a chance to settle and they are in the presence of large copepods and zero-group gobies. Recruitment failure in gadoids or any species can spell the end of a once thriving population, and a lot of researchers have examined both the extent of the failure and the impact on the ecosystem. And concurrent recruitment failure in gadoids, which includes our cod, and changes in the plankton community were found along the Norwegian coast after 2002, which actually could have affected the study area of this project. Recruitment could also be affected by human activities, as demersal fish catches have been dominated by immature fish, and cod especially showed changes in size, meaning that fishermen are now catching cod that are getting smaller and smaller. Now let's talk about the stereo BRUV system, which stands for Baited Remote Underwater Video. It's a very useful tool for implementing studies in benthic habitats, especially protected areas. The rig itself is a metal frame, which is called the base bar, with two GoPro cameras mounted to give you left and right video. Down the middle of the rig is an arm that at the end of that orange bit, there's a mesh bag that contains bait, which for this study was frozen Atlantic mackerel. So now that we've covered the background information, what was actually the whole point of this study? Well, we actually had a lot of questions and objectives here, so bear with me. Our first goal was to gather baseline biological data. We wanted to create an inventory of fish assemblages. We wanted to scope for features of habitat and substrate that is conducive to harboring young of year cod. And we wanted to determine factors with explanatory properties regarding occurrence and co-occurrence of cod recruits and juveniles of other species. We had a lot of research questions to tackle. Does the presence of cod have an impact on diversity? And does it have an impact on overall abundance? What specific factors predict cod presence in the environment, with the factors being habitat type, depth, and diversity? Which factors influence the abundance between cod presence, habitat type, and depth? And we wanted to compare several things from northern group of stations and the southern group of stations. Is there a difference in length measurements, diversities, and abundances? And also, is there a difference in abundance between the northern group, the southern group, and the middle station that I'll explain in just a minute? So here is a map that was constructed from the latitude, longitude data from the BRUV drops. So up here, we have the northern group of stations. The southern group is down here in Fado National Park. And then we have this middle outlier station, and I'll explain why later that's important. So these data were collected in August of 2017 over a 3-day cruise. There were 35 stations visited in Fado National Park, but only 18 ended up having usable footage. The protocol was to lower the rig onto the seafloor and record 1 hour of video per station sampled. Now for each camera, calibration files had to be set up so we could actually measure our individuals and event measure. The way these files are written, it's based on the principle of photogrammetry, which is the science of determining measurements from photos. It's based on triangulation and very similar to how human eyes process depth perception. And it is important that you get measurements from several angles to make this process work, and this is nicely illustrated here. If you only do it from one point, okay, great, you know a lot about this one little point, but you don't know really anything about the object as a whole. But once you go from multiple angles and multiple points, now you have a clearer picture of what this 3D object looks like. And that's how Event Measure gives us the measurements. So Event Measure is a very cool program. And the protocol for it was all species that were visible in the video had to be identified, and then the individuals were counted. To be identified and included, it had to be fully in the frame, and you had to identify it to at least the family level. And then each was measured if it was possible to do so, if the organism is visible on both cameras. So he can't be fully in one and half out on the other. For densely populated stations, especially if there was a large school that hung out in the same spot for a long period of time, a few key frames were selected with the greatest number of individuals, the highest number of species, and the most potential to be measured. I implemented this into the protocol for two reasons. One, we're not really concerned about double counts, and I'll explain why later. And two, I wanted to finish this project before I died of old age. So here's a screen grab of what Event Measure looks like. It's a very useful program that can give you data in different formats. You can see points, you can see length measurements, you can check abundance. And in this particular one, we have a group of juvenile Atlantic cod with an edible crab. And this was definitely one of the coolest stations I looked at, and a very good example on how to measure them. So this is a school of safe. And once you measure it, Event Measure will tell you you type in all the biological information, and then Event Measure tells you the length of the fish, how precise was the measurement, and most importantly for my changes to protocol was the Z coordinate, which is the horizontal distance from the camera. And this little number is important because I made the call to exclude any individuals that were past 2 meters from the camera, just because it was harder to identify, and I wanted to keep it scientifically, integrity. And I just want to show you a few cool ones. Here's an edible crab, Cantopagaris, and this is a juvenile Atlantic cod that we fondly named the bully cod because he was a total jerk and did not allow any other fish to have the bait. We have an edible crab again, more juvenile cod, and this guy was my favorite. He is the greater weaver, Trachonus draco, and he was super duper cool, so he had to go in the presentation. So before we continue, I would like to define max N because it's a big part of this project. So max N is a proxy term for abundance that's used in grub studies. It is defined as the maximum number of individuals for a given species counted within the field of view at the same time. It's a great method for grub studies because it avoids double counts, which is what I was saying earlier about densely populated areas. We calculated by using the number of fish within the focal distance of the camera and then summing those counts. And then after the data was cleaned up a little bit in Excel, because RStudio is very particular what you put in it, I began the data wrangling section. First, I added a column to reflect the number of particular species in every frame. Now, I did this really to condense the data to make it more workable. Because, for example, if frame 47 had 18 fish, it's going to pop up 47 times, and that's a nightmare to analyze. So instead, by condensing it into this count data set, now we have frame 47 has 8 cod, 3 gobies, and a flatfish, which makes it much more easier in that format. I also added a presence-absence factor and a diversity index column, which was filled out by the separate data frame I made containing Shannon-Weiner diversity index values. So once I merge all this together, I have a complete count data set. And I took this data set to do 2 chi-square tests, one to determine if cod presence has any impact on the presence of other species, and one to see if it impacted max N. Then I constructed linear models. So the first one wanted to examine diversity in terms of cod presence and absence and cod counts. So the way this equation is structured, our response variable, what gets spit out, is the diversity. Our predicting variables are cod presence and cod count. The next equation is factors that predict cod presence. So with cod presence as our response, with predicting variables of habitat type, depth, and diversity index. Then it was factors that had the strongest influence on max N, with max N as our response, and cod presence, habitat type, and depth. Then we did 2 sample t-tests to determine if any statistically significant differences between the north and south stations existed for length measurements, diversity, and abundance. Now, after I did that, I noticed that the middle outlier station I pointed out on the map had the highest max N, way higher than the other ones. And I was very curious to see how that interacted or related to north and south. And while 2 sample t-tests are great for 2 samples, they're only good for 2 samples. So for a third, I decided to go with a one-way ANOVA. But there are assumptions that you need to meet when doing a one-way ANOVA. The first assumption is each sample originated from a normally distributed population. The Shapiro-Wilks test suggested that it may not be, but the QQ plot showed that most of the points fell along the line. There's just a couple outliers at the bottom and the top. And a one-way ANOVA can actually withstand deviations from a normal distribution if your sample size is large enough, which mine was. So I decided we're within the threshold and we can make it work. The second assumption is each sample is independent of all other samples. There's no formal test to check for this, and we can't be 100% certain because, fish move. So they may have gone to another station, but because our sampling procedure was so randomized, we can be confident that the second assumption holds true. The third assumption is that the variances in each group is equal, which I checked out with box plots and Levine's test. Because Levine's test is less sensitive to slightly abnormal distributions, which is what we discovered in assumption 1. And the test said that there may be significant differences, but the box plot showed very minimal, and the middle one was just a little bigger. So at this point, it's up to the researcher whether they want to be conservative and do like a Kriskus-Wallace test, which is the non-parametric version of ANOVA. But I decided that everything was within the threshold and wouldn't derail ANOVA, so I proceeded with this equation here. And there's our box plot. So you can see north and south, very minimal difference. And just because the middle had such a higher max N, that's probably why Levine's test was not too happy with me. So now we get to the results, which is the fun part. So first, before I started doing the R work, I wanted to see what was the most abundant family, just to see if we're on the right track examining cod. And turns out, yes, Gattids was the most predominant family. That includes cod, whiting, pollock, saithe. Labrids, which are wrasses, were number 2, followed by Fortunidae, which is shore crabs, flatfish, big crabs like that edible crab in the pictures, gobies, and then some other families that were so minimal they're not very visible on this pie chart. So now I had the most abundant family. What was the most abundant species in the most abundant family? Cod won by a landslide, which is honestly not that surprising. Then I calculated density plots for overall picture across all species and in every individual species. Unfortunately, I can't show you every individual species because we'll be here for a week, so this is just the general picture. So overall, across all species, the average length of the fish was 118 millimeters, so pretty small. The linear models gave us some really interesting plots, so I'm going to show you 3. On all 3, your y-axis here is the probability of seeing a cod. On the x-axis for this one is the habitat type. So this tells us that it is more likely to see cod in a sandy shell habitat, but more variance in a rocky mixed. This x-axis is depth in meters, and this one tells us that it is more likely to see cod in shallower waters less than 20 meters. And you can see a pretty drastic change after 20. This x-axis is index values for the diversity index, and there is a relationship, although weak, between cod presence and diversity. So all the testing came back and showed us a significant relationship between cod presence and diversity, so we accept the alternative hypothesis that cod presence impacts diversity. The linear model determining if cod presence leads to different diversities suggested a negative effect of cod abundance on species diversity. Then for the generalized linear model that we did to determine factors that predict cod presence, we ended up selecting the best fit model with the AIC values. And we made 4 different flavors of this model to check it. So the global model had all factors: habitat type, depth, and diversity. The next three excluded one. So one excluded habitat, one excluded depth, and one excluded diversity. We just wanted to see how these compare, so we have the best fit model. The best fit model ended up being the global model that had all of the factors involved, with sandy shell habitat having a positive effect on cod presence. Depth had a negative effect on cod presence, which makes sense with the plot we just looked at, where they prefer shallow waters, indicating that cod will be present with higher max N counts in shallower stations. Diversity also showed a negative effect on cod presence, suggesting that fewer species other than cod were registered at stations in which cod were present, meaning if you see a cod, there's probably not many other species there. There were no significant differences in terms of the length measurements from north and south. They had pretty much the same average, and the abundance had the same average, but there was a significant difference in diversity, and there was higher diversity in the southern group of stations. For the max N, the chi-square test said that there was a significant relationship, and there is an impact of cod presence on max N. And the linear regression model to see which factors had the strongest influence on max N said a rocky mixed habitat was the most influential factor with a positive effect. So the ANOVA I was talking about earlier just tells you if there's a difference. It doesn't tell you where the difference in means are. It just says yes there is or no there's not. The ANOVA test came back with yes there is a significant difference. So then the Tukey test always tells us what specific differences there are. And what was very intriguing was north and south wasn't different from each other, but they were both very different from the middle station. So what do all these, that's a lot of numbers. What do all these numbers actually mean for the big picture? Well, we discovered that the fish species assemblage is dominated by relatively small-sized species and juveniles of larger species. Young of year juveniles was the most abundant age group and year class for the cod, and species that are known to be of a larger size, like pollock, they can get pretty decently sized, they were also pretty small, which supports the conclusion of a juvenile-dominated community. And nearly all cod observations were juveniles. After filtering out the 2 meters, there were still 534 cod, and out of the 534 cod, only 18 were adults. The diversity between the north and south stations were different, which is actually really interesting because the literature stated that the juveniles tend to settle away from the open ocean in more sheltered areas. The southern group is more toward the open ocean, so that doesn't follow the literature. This may be due to that the southern stations are in Fado National Park, which is a marine park, and that may provide better resources and protection. But given this was kind of a small-scale project, it warrants further investigation. And while north and south weren't different from each other in max N, they were very different from the middle station because the middle station had a top max N of 69. So that also warrants investigation as to why does that middle outlier station, why is it so appealing to the species? Personally, my favorite finding from this was that the presence of cod wasn't significant. It's the actual number of cod that influences the diversity. And sandy habitats and depth were the most influential figures in cod presence. Now, why does this make sense? Well, going back to the literature review, it stated that cod, especially juveniles, utilize different depths in sandy habitats during warmer temperatures. And if anybody's been in the waters here in August in Norway, it's like a bathtub, so that tracks. Juveniles are less sensitive to warmer temperature than adults, so that explains why we saw so many juveniles. And the growth of juvenile cod is slower in the summer, which explains how we not only saw juveniles, but small juveniles. This has so many implications for management agencies. Agencies have been criticized in recent years for not incorporating recruitment stages into their protocols. They tend to focus on the adult fish stocks, which doesn't really make sense because the juveniles turn into the adults that go into that fish stock. And if you don't have any more juveniles, you're not going to have adults and you're not going to have a stock. And the shallow habitat throughout the park from north to south is a nursery habitat for many species. So that is a game changer within itself. And if these juveniles aren't protected, and for example, their habitat here is not protected, the stock can collapse for any number of reasons. There could be genetic collapse from inbreeding, or another case where populations have been known to mature at younger ages and smaller sizes recently, which can lead to juveniles with decreased fitness and other complications. This has been seen in many places. One of my favorite papers talks about how older, larger rockfish give birth to tougher offspring that can survive rough conditions and starvation longer than offspring from smaller, younger females. So we really need those big old girls in the group to keep the stock healthy. Another thing that management can think about is the source-sink dynamics, which is an ecological model to explain how the same species can have different populations in two separate habitats. The source is a high-quality habitat that encourages growth and is practically overflowing. The sink is a low-quality habitat where the decrease in population is faster than the increase. The sink is not sustainable within itself, but it can be made sustainable by migrating individuals from the overflowing and flourishing source habitat. If one were to do a population genetic study, that could determine where the juveniles are occurring from, which means the spawning site would be the source, and where they could possibly go, which makes the nursery habitat the sink. And this would also provide really useful information about home range and connectivity of subpopulations. And for this to work, there would need to be extensive protection for both habitats with a possible network of protected areas. And this could also tie into the actual eggs and larvae themselves, because a lot of literature has also stated how currents affect the egg dispersal, which this would help figure out where that spawning site source is. Baseline monitoring studies like this are very important to scientific knowledge. It gives you a place to start. It provides a foundation for future studies to build upon, and it highlights specific questions that are yet to be investigated and which gaps need to be filled in. And future studies need to be mindful of the shortcomings of baseline studies and aim to fill in those gaps. For example, the presence-absence data in my study. Presence-absence is very useful for a lot of questions, but it does have a tendency to over-exaggerate the presence of more rare species like the greater weaver and the spotted ray I saw. So if you look at my data, you would think there was a ton from the presence-absence, but that wasn't the case. So this is some things that future studies need to keep in mind. So in conclusion, BRUVs may be used in temperate and relatively turbid conditions. BRUVs are highly useful in assessing young of year juveniles and their interactions, not only between other species, but their environments. And this is a crucial step for management agencies that are in the process of establishing and maintaining viable fisheries and marine protected areas. Future work should include more shallow waters. The data were collected 10 meters or lower. They didn't do 0 to 10 just because they have different equipment that can do that. But it would be interesting to see this method in shallower waters and what data comes out of that. Future work should also focus on juveniles of all economically and ecologically important species, because stronger protection of juveniles is going to be the key to both fisheries success and the health of the surrounding environment, and tie into maybe new recruitment protocols and management strategies. And because physical factors such as temperature can really influence habitat preferences, that should be toward the top of the list. And because the Oslo Fjord is so unique being in the Skagerrak Sea, the physical composition of the environment and the waters themselves should be examined. This is a lot of work to do, but it would be absolutely instrumental in keeping the environment healthy. I have a lot of people to thank. First, I'd like to thank my supervisor, Evan Mullins, for the support and guidance and having me up at the lab and showing me how to use all these really interesting programs. I want to thank Anna and Kim for being on my panel today. Thank you for agreeing to examine my presentation. I really want to thank Tova, not only for helping us pick a thesis, because that's a big step, but also just for being so supportive during this pandemic. grad school makes you have meltdowns outside of a pandemic. So Tova checking in with us and making sure we were okay made a big difference. So thank you so much for that support. For statistical help, Dr. Ali Kramer of University of California, Santa Cruz. She was my original research mentor during my bachelor's, and she is my Jedi Master and taught me how to science. And Marta Sotolan here at Ouya helped me navigate the turbulent waters of data, and I could not have processed that data without them. So thank you so much. I have so many friends and family. A bunch of them are on here right now. I don't have time to name all of them, but thank you so much for getting up at 4 o'clock in the morning, supporting me, making sure I was eating, letting me bounce ideas off you, reading my paper, giving me feedback, letting me have meltdowns. Couldn't have done it without you guys. And I definitely want to thank, last but never least, my amazing partner I love so much, Ian, and his parents, Diana and Richard. Your unconditional love and support was the catalyst for this accomplishment. And I would not be here today without you guys. There's all my references. There were a lot, but I promise they're on the paper. And now I shall take your questions.